Vanakkam. Welcome to this course on classics in neuroscience. In this series of videos, we are looking at cell biology, specifically the nature of the synapse. We have been looking at the chemical synapse. In this class, we will be looking at a background on synapse study. We try to identify or we try to present the points of view regarding who discovered the synapse, and we discuss the early work on neuromuscular junction. So, in our previous videos we saw that the first electron microscopy images of synapses were reported in 1954 independently by Sanford Paley, George Palade and Eduardo de Robertis. So, in the 1954 meeting of American Association of Anatomists, George Palade and Sanford Paley presented two short communications, two brief communications. George Palade presented the electron microscopic observations of interneuronal and neuromuscular synapses. He also described observations in the cerebellar cortex and in the medulla oblongata. He observed visualization of mitochondria, vesicles in axon endings and a dense plasma membrane at contact points. Because there is a dense plasma membrane, there is essentially a discontinuity of protoplasm. There are two cells, each with their own protoplasm, and there is a dense plasma membrane separating the two and a gap between them, suggesting that there is a clear lack of continuity. So, they studied neuromuscular synapses in rat skeletal muscles, and they showed how there were similarities between the neuromuscular junction or the neuromuscular synapse studied in the rat skeletal muscle and central synapses. Whereas Sanford Paley, in his report, where he said he was assisted by George Palade, in the previous paper George Palade was assisted by Paley. So, they were both first and second author, exchange authorship maybe. He presented an abstract or a short communication called electron microscopy study of the cytoplasm of neurons, and he reported finding Nissl substances that's shown here, Nissl bodies composed of members and granules, and he mentioned Golgi body. Remember, until 1949 people were saying that Golgi body is an artifact. So, for the first time he reported that Golgi body is something that he is able to observe using electron microscopy, dendrites and the similarity of dendrites and axons due to some fixation limitations. In the same meeting, that is the American Association of Anatomists in 1954, Eduardo de Robertis presented his own brief communication or his own study titled "The Changes in the Synaptic Vesicles of the Ventral Acoustic Ganglion After Nerve Section." He focused on this area in this region because he said that it is possible to visualize the nature of synapses after destroying the cochlea in guinea pigs. So, he described synaptic vesicles, mitochondria, endoplasmic reticulum in the presynaptic nerve end. So, he noted there is contact between the synaptic membrane with the postsynaptic cytoplasm, and he suggested that there are degenerative changes in the terminal after nerve damage, but this was not found in the other neurons. This suggested that degeneration of one neuron does not pass on to another neuron, because if there was continuation as demanded by reticularists, then degeneration must also continue from one neuron to another neuron, from one part to another part. But that does not seem to happen. Degeneration of one neuron causes death or degeneration of that neuron, but that does not pass on to another, providing a strong evidence for the neuron doctrine. But this is being currently questioned because there are neurodegenerative disorders that pass from one cell to another cell through other means not discussed here. There is still discreteness, so neurons are discrete with each other, but it is possible for neurodegeneration to happen in multiple neurons through communication between neurons. That is a possibility that we have now discovered or we are now convinced about, but that was not obvious at that time. In any case, that is a different story. Here what is suggested is that death of one neuron does not automatically cause death of another neuron, or degeneration of one neuron does not cause the degeneration of the neighboring neuron, suggesting that neuron doctrine is the most possible, most plausible explanation of the situation. And he provided structural evidence, morphological evidence for synaptic structures and how transmission, information transmission happened chemically between neurons. These early brief or short communications were quickly followed up by with full papers by both D. Robertis and Bennett and Paley in 1955 and 1956. And in 1956, Sanford Paley defined some key features of the synapse. What are these features? That is close proximity or closely limited presynaptic membrane and postsynaptic membrane, presynaptic and postsynaptic membrane or dendrite or whatever membrane, and there is a cluster of mitochondria. Importantly, vesicles that were later theorized to contain the chemical messengers or the neurotransmitters were found in plenty in the presynaptic terminal, but not the postsynaptic terminal. A clear absence of protoplasmic continuity, absence of protoplasmic continuity, that is, there is a clear gap between one cell membrane and another cell membrane, that is there is a protoplasmic discontinuity, that these are two different cells, providing an impressive and an extremely strong evidence in support of Ramoni-Cahal's neuron doctrine, that is absence of protoplasmic continuity, thus finally providing convincing evidence in support of neuron doctrine. More than 60 years after the first formulation of neuron doctrine, finally there was this convincing evidence. But how is this vesicle communicating with the neuron or the cell on the other side and the postsynaptic cell? It was not clear, and the first description of this was by the theory that was developed by Edward D. Robertis in 1959, where he theorized that vesicles fuse with the plasma membrane of the presynaptic cell and release the neurotransmitters or the chemical messengers. This process is called exocytosis. Through this process of exocytosis, this was the first theory of how perhaps this chemical messenger can communicate with the postsynaptic cell. The question of who discovered the synapse, that discussion continues to be controversial or contentious. One thing is clear, that is that the discovery of the synapse was entirely dependent on the development or the advent of electron microscopy technology. So, whoever had access to electron microscopic technology was able to demonstrate this. In this case, both Sanford Paley and Eduardo de Robertis both had access to electron microscope. So, in 1988 Sanford Paley was invited to give the history of neuroscience lecture at the Society for Neuroscience annual meeting. This talk was sponsored by the Society for Neuroscience Committee on History of Neuroscience. So, in that talk and the dinner that followed, Sanford Paley was very moved and very touched and described his feelings about how he was the first to observe synaptic vesicles. He also described this in his 1992 memoir. In particular, he placed a very high emphasis on the quality of the images that he was able to produce, to say that I mean, it is not just finding something, but the quality of the images that were much higher. So, for him, his priority, he was among the first to demonstrate with high quality images. Throughout this talk and the discussion, he never mentioned the work of Eduardo de Robertis. So, Eduardo de Robertis and Bennett, they claimed that Eduardo de Robertis provided the first proper description of the synaptic vesicles and space between the neurons in the synapse in 1955. Both Sanford Paley and Eduardo de Robertis are acclaimed scientists with a great love for their art or their science, their craft, and teaching. Both authored textbooks, several papers. It does not matter who contributed or who was the one who discovered. The point is that such was the quality of work that was done in the 1950s. So high was the quality that both were equally good and both were real doyens of this field of neuroscience. So, shown here is a picture drawn by my student with her own hand, I think, showing essentially the visualization of synaptic vesicles, and one vesicles releasing the neurotransmitters and receptors on the postsynaptic side, etc. Later, the work done by George Gray in London using appropriate fixation methods showed that indeed there existed two types of synapses. Type 1 synapse, those having round vesicles, and type 2 synapse, those having vesicles of different shapes, pleomorphic. It was suggested that type 1 synapses were excitatory and type 2 synapses were inhibitory, and also the membrane density in the type 1 synapse was not symmetrical or was asymmetrical, and the type 2 synapse it was symmetrical. This discovery or this distinction was critical in future to identifying or to studying of neuroscience or the nature of inhibitory and excitatory circuits in the rest of the central nervous system in the forthcoming decades, that is the years following the late 1950s, 1960s, 1970s, etc. In addition to these type of synapses, there was also tight junctions, that is opposed membranes, not opposed as in with O, but rather opposed. What is the meaning of this word? Check, Google is your friend. Opposed membranes held close to each other, tightly held with each other, forming barriers to prevent extracellular substances from entering the neurons, essentially forming barriers such as the blood brain barrier, for example. Now, let us return to where we originally began our discussion of the synapse, remember an important model system for the study of the synapse was the neuromuscular synapse or the neuromuscular junction. So, neuromuscular junction was a critical model system, and it was assumed that this was similar to other synapses. This neuromuscular synapse essentially represented how the other synapses also worked. One of the pioneers of the study of neuromuscular junction was J.D. Robertson. He studied how the neuromuscular junction functioned in the reptiles. So, essentially the early study of neuron and the muscle membrane and the junction between them in reptiles, and he discovered the double leaflet structure of the muscle membrane, and he found that neuromuscular junction comprised of a presynaptic neuronal membrane and a relatively more convoluted muscle membrane, and he identified multiple clusters of vesicles that were later called as active zones, for example by Dean Kotia and Heuser and others. And also the release of one vesicle into the synaptic cleft caused one minimal amount of change in the postsynaptic excitable cell, in this case the muscle cell, called as miniature end plate potential. This is the smallest change in the potential of the postsynaptic muscle cell that can happen. Remember, it is not the change that is made by one acetylcholine molecule, but rather when one vesicle is released, the change happens due to all the molecules, acetylcholine molecules found in that vesicle. So, the change in that voltage in the postsynaptic cell is, let us say, some small change which is called as MEPP, miniature end plate potential. The idea that vesicles release neurotransmitter as quanta was first discovered and described by Del Castillo and Bernard Katz. That change in this potential can happen only by quanta, by multiples of this MEPPs, for example. And also uniquely, there was this basal lamina found in the synaptic space that is unique to the neuromuscular junction, but not found in other synapses in the central nervous system, for example. And also later people were able to localize acetylcholine esterase. Remember, we discussed acetylcholine esterase in our previous discussion, and also in the receptors of acetylcholine in the postsynaptic folds. So, this structure of neuromuscular junction, or the morphological description of the neuromuscular junction, aligned very well with the simplified central synapse, or the morphology of the simplified central synapse. This helped provide solid evidence for the concept of structural synapse or morphological synapse, providing convincing evidence to support the neuron doctrine. We will be discussing the work of Bernard Katz in future chapters. In future videos, we will be discussing the work of Bernard Katz. I did ask you to check about this person, Bernard Katz. I have mentioned more than once how he escaped Nazi Germany and lived in A.V. Hill's lab and went on to win the Nobel Prize for his work on identifying the nature of synaptic vesicles. With this, we come to the end of this video. In this video, we discussed the nature of synapse and how or who all contributed to identifying the synapse, and the contention, the controversial question of while Sanford Paley claimed that he was the first to visualize the synapse, Eduardo D. Robertis and Bennett claimed that they were the first. They both were almost contemporaries, almost they did this perhaps almost simultaneously. Sanford Paley emphasized the quality of his images to get that priority, and in his talk he did not discuss the parallel work of Eduardo D. Robertis, something to keep in mind, and the work of Robertson on neuromuscular junction, and the work of Bernard Katz on vesicles. With this, we come to the end of this video. Thank you very much for your attention.